Hi, my name is Syed Imam, and today I'll be talking about familial Creutzfeldt-Jakob disease. This is a basic form of the protein that was obtained by a PDB file, and what they basically found out after years of research is that there's a GPI anchor over here, multiple carbohydrates over here that provide a stability to the molecule. Alongside that, there are multiple alpha helixes that don't really show very well on this, to me at least, and later on there's a better image of that, and there's also some disulfide bridges, and there's also a couple of beta sheets in there as well that produce stability. With the background that I obtained from nih.gov, it's a transmissible spongiform encephalopathy, or TSE. It is a very rare disease which occurs in approximately 1% out of 1 million. It is neurodegenerative and it's a fatal brain disorder. In US, there are approximately 300 cases per year, and majority of the cases die within a year of the disease occurring. And as shown by the symptom, it occurs it's related to cerebral ataxia or cerebral function loss, and so that tells us that it's basically based around neurons. So with the normal protein, majority of them, as I talked about earlier, a large amount of them are present in brain, and they're mostly tethered to the outer membrane of neurons via this GPI anchor. The protein itself is 209 amino acid long and rich in alpha helices. Over here you can see those three alpha helices that I was talking about. There's the disulfide bond that provides stability, and the N-linked carbohydrates that also provide additional stability to the whole molecule, allowing it to function properly. With the abnormal prion protein, it actually causes a conformational change where the abnormal is actually rich in beta sheets, and it is this abnormal form is usually found within vesicles which contain red blood cells or lysosomes, because they utilize the energy available in these cells to denature the protein and then turn it into beta PRP that then directly has irreversible propagation to a PRP-SC, or it has nucleation with a PRP-SC seed until there's an irreversible propagation to PRP-SC. With familial specifically, there is a gene mutation in the PRNP gene, which I'll talk about in the next slide. It's basically a misfolded PRPC monomer that turns into this PRP-SC. This molecule is actually more stable due to the fact that there's more monomers available, so there's many more bondings occurring, hydrogen bonding, LDF, and so on. And this is also an explosive autocatalytic reaction due to the fact that there's some PRPC that is shown in PRP-SC. This model that I obtained doesn't really show that very well with the PRP-SC, but later on I'll talk about the structural biology that does contain a model that shows this autocatalysis that occurs with PRPC to PRP-SC. So the genetic mutation that leads to PRP-SC forming rather than PRP through the PRNP protein that generates the PRPC protein, and it's called a codon 129 polymorphism. So someone who has this codon 129 polymorphism can be a methionine homozygote, methionine valine heterozygote, valine homozygote. And what this study by Pucci et al. Found out was that people with the methionine homozygote zygous are 70% susceptible to this disease, 11% in the case of the heterozygotes, and 19% for the valine homozygotes. And then they found the cleaving site, which changes depending on methionine or valine or heterozygotes, which is amino acid number 82 in the case of methionine, or amino acid 97 in the case of valine or heterozygotes. And I wasn't able to obtain the whole article because of a paywall, but from what I understood from Swansea University's medical schools module on prion diseases is that this cleaving is the reason why there's that beta sheet change that occurs, and alongside that they also found some of that resistance that's found with PRP-SC or PRP scrappy, is that the protein is undigestible by a lot of these enzymes that usually degenerate these proteins that are not needed in the body or are harmful to the body. So the big thing that was found by Collins et al. Is that depending on how quickly the amyloids or the aggresomes that these are capable, the abnormal protein is capable of form, the disease occurs based on that. So what they found was that these aggresomes form together amyloids that form fibrils, and they go around converting those normal proteins into abnormal prion proteins. And I'll talk about these mechanisms, what we have understood through research, later on in the presentation. The thing that they focused on was the methionine 129 PRP, the methionine homozygous, and the valine homozygous. And what they found out was that the methionine 129 homozygotes have a lag time or incubation period of 6.8 hours, shown right over here. And the V129 or the valine ones have this very long lag time of 14.5 hours, which shows why has that increased susceptibility, because they can increase in population so much faster. What they found out was alongside that was that V129 polymerized quicker, but M129 was more prone to unstructured oligomers. And what that means is that because they're unstructured oligomers, it's more capable of creating that nucleation site so that it can turn PRPC into PRP-SC much easier, because there's that additional availability of energy to form more stable bonds. And alongside that, in that same study, they also found some additional information about resistance that I was talking a bit about earlier. And what they found out was that basically beta PRP is basically undigestible, whereas alpha PRP, or increased amount of alpha helices, are much more much easier to be broken down by proteinase K. And proteinase K, what they found out was that it's optimum around pH 8.0, and it seemed to digest proteins preferentially after their hydrophobic amino acids interaction. So in this one, you can see that it's being done at the optimum pH, but this one, the pH isn't exactly optimum. It's at 4.0, which is much less, and I'll talk about it a bit more later on, because there was a research that basically found out that these beta formations occur more so in lower pHs than higher pHs. So it shows why these Collins et al. Decided to go with 4.0 versus 8.0. But to compare and say that, it's not because the protein isn't working properly, they also digested alpha PRP at the same 4.0 pH. And alpha PRP, as shown in these two, they were still digested, whereas beta PRP is present over here, shown by these immunoblots. So going into the biochemistry of PRP itself, it has a folded C-terminal, a largely unstructured N-terminal. The N-terminus actually contains 4 glycine-rich octapeptide repeats that have a high affinity for copper, and they also have 2 potential sites for N-linked glycosylation, which was shown in the earlier image. It showed those carbohydrates attached to these helices, and that occurs at these 2 sites that they found out. And there's also that disulfide bond that the earlier model showed as well. With normal PRP, what they found out was that it's monomeric, as in it's built from these bunch of monomers, and there aren't a lot of them, so it's still relatively monomeric. It is very sensitive to proteinase K, and it's very soluble in pretty much any non-ionic detergent. With pathogenic PRP, it's actually insoluble, resistant to PK digestion, as shown in this study, and it's aggregate in nature. And alongside that, what they found out was that based on those genetics that I was talking about earlier over here, was that this additional aggregation might actually be occurring because of those beta sheets that occur due to this polymorphism. In the version, there is this slight polymorphism. There is slight amount of beta sheets, but what they found out later on was that there's also a change in codon 200 where glutamic acid is actually turned into lysine, and lysine is prone to making more beta sheets, which showed why even though methionine would form alpha helixes, it still produces a lot of beta sheets, which could be potentially because of that lysine available. Whereas with these heterozygotes and valine homozygotes, it's pretty clear because valine is a beta sheet former, but methionine is a alpha helices former. So if methionine majority is removed, then you're going to have more beta sheets than alpha helices. So going forward, we're talking about the prion protein conversion mechanism, because even with familial, this is very important because of that long incubation period. So obviously there is some high protein conversion going on still, even though it is a genetic mutation. And what they found out through several studies, I read close to 15, and I chose these ones because they had the most revolutionary effect on the research itself. The first one being the heterodimeric refolding mechanism, and what this one found out was that PRPC, which I'm going to refer to as PC from now on because I'm going to be saying this so much, is much less stable thermodynamically than PRPSC, or referred to as PSC, so from this point on. And since it's less stable, this reaction will move forward towards the more thermodynamically stable form. And what this study also found out was that this conversion is actually kinetically limited. So this was basically thrown out because of this idea that, it's kinetically limited, whereas the actual thing when it happens in human bodies is it's very kinetically powered, and it's very spontaneous. But what they did find was something that was confirmed later on, was that there's a formation of a heterodimer between PC and PSC that uses the monomer template of PSC to convert PC into PSC. And it was shown later on that there is in fact a monomeric intermediate, and this was indicated by kinetic studies, hydrogen deuterium exchange, and high pressure spectroscopy experiments. And what they found out about that heterodimer or monomeric intermediate is that it is highly hydrophobic, and its nucleic acid backbone is actually exposed. So this allows for the increased propensity of intermolecular interactions, and it also increases the stability, so that monomeric intermediate provides the additional energy needed to turn PC into PSC. And later on, the agrosome idea was confirmed by this study, where they used protein misfolding cyclic amplification, or PMCA. And what they found was that they used successive rounds of sonication and incubation, so basically sound energy into letting it recover. And what they found out was that there was indefinite amplification of PC to PSC. And what these previous studies couldn't do was that they couldn't cause the disease, which showed that they needed more of them. But this one confirmed it, because when they took that PSC, it caused disease in animals. And so alongside that, what they were confused about though was that if it was occurring due to the fact that there was increased loci for substrate attachment, or there was just additional energy being produced. And after that, they did a recombinant prion protein study where they used bacteria to obtain prion proteins. And what this study showed, as I was talking about earlier, is that the prion protein actually functions better at mildly acidic pH, and it converts better at that mildly acidic pH. But the problem with this one was that it was non-autocatalytic. Lastly was the nucleated polymerization mechanism, which showed that oligomers needed to be created because they act as a nucleus for these monomeric conversions from PC to PSC. And what they said was that PC gets deposited within the nucleus, and the nucleus provides the energy to help it adopt the new structure. And the evidence of that was when they put PC with protease resistant PRP, even though a lot of PSC was not pretty present, PC still turned into PSC. So this is what I was talking about with the structural biology and the autocatalytic. You can still see over here there are still signs of PC in the PSC molecule. Over on this side, these are just a bunch of yeast prions that they use to study the mammalian prions, and this is where I was talking about additional stability with the PRPSC. What they found out was that these beta sheets are stacked in such a way that they continuously form back and forth layer between each other, going like this. So there is those anti-parallel beta sheets are producing bonds over here, but there's also Linden dispersion bonds between these sheets that produce additional stability. So overall, there with this familial CJD is that PRPC is rich in alpha helices, whereas PRPSC is rich in beta sheets. PRPC attaches to lipid rafts, but the other one forms aggresomes that infect and cause apoptotic death of neural cells, while PRPC protects those cells. One is digestible by PK, the other one is non-digestible by PK. With familial, it is a codon 129 polymorphism of the PRNP protein, which shows susceptibility to disease, and methionine polymorphism is a much bigger indicator than valine or methionine heterozygous patients. And these multiple theories that I talked about showed us how close to a perfect image of how things function, and what they basically found out was that there needs to be some form of a polymerization nucleation mechanism, so it needs to create those oligomers. It is only protein only interactions. There's no DNA RNA involved. The protein just changes conformers while interacting with other proteins. There's some sort of a monomeric intermediate. There's thermodynamic stability, which provides the energy to push the conversion along, and the whole reaction is autocatalytic. And that's pretty much it. Thank you for listening.